Imagine two patients, same cancer, same stage, same treatment. One walks out of the hospital 5 years later, cancer-free. The other doesn't. The only difference, whether their cancer decided to spread. Metastasis, when cancer cells break free from the original tumor and invade other organs, is responsible for roughly 90% of all cancer deaths. And here's the terrifying part. Doctors still cannot reliably predict which tumors will spread and which will stay put. But in March of 2026, a team of researchers in Switzerland may have just cracked this code wide open. The team was led by Professor Ariel Ruiz, Altaba, from the Department of Genetic Medicine and Development at the University of Geneva. For years, scientists have treated cancer like chaos, anarchic cells mutating randomly, spreading unpredictably, defying all logic. But Ruiz, Altaba's team, discovered something that challenges everything we thought we knew. Cancer isn't random at all. It follows a biological program. Think of it this way. During embryonic development, your cells follow an incredibly precise set of instructions to form organs, tissues, and systems. Those programs are supposed to shut down after development is complete. But genetic and epigenetic changes can reactivate these dormant programs in adult cells, and when they fire up in the wrong context, they drive cancer formation and metastasis. In other words, cancer isn't a rebellion. It's a distorted echo of your own body's earliest instructions. To prove this, the UNIGE team did something remarkable. They isolated individual cancer cells from colon tumors, cloned them, and grew them separately in the lab. Then co-first authors Arwin Kanad and Aravind Srinivasan tested each clone's ability to migrate through biological filters, both in Petri dishes and inside living mice. They measured the activity of several hundred genes across roughly 30 cloned cell lines taken from 2 different colon tumors. What they found was not scattered noise. It was a clear gradient. Cells with certain gene expression patterns were highly migratory, eager to spread. Others with different patterns stayed localized. The relationship was remarkably consistent. Even more surprising, the risk of metastasis couldn't be determined by studying a single cell. It depended on how groups of related cells interacted within the tumor. Armed with these gene signatures, the team built an artificial intelligence system. They called it MangroveGS, short for Mangrove Gene Signatures. Unlike previous tools that rely on 1 or 2 biomarkers, MangroveGS analyzes DOZ and S, even hundreds of gene signatures simultaneously. This makes it resistant to individual variations between patients. The results, nearly 80% accuracy, predicting whether colon cancer would metastasize or recur. That significantly outperforms every existing prediction tool currently available to oncologists. But here's where it gets really interesting. The gene signatures they identified in colon cancer also predicted metastatic potential in stomach cancer, lung cancer, and breast cancer. Same biological program, different organs, one AI model that sees the pattern across all of them. The study was published in Cell Reports in late December 2025, one of the most prestigious journals in biology. So how would this actually work in a real hospital? A doctor takes a tumor sample from a patient. The cells are analyzed. Their RNA is sequenced right there in the hospital. The system generates a metastatic risk score, transmitted securely through an encrypted portal called the Mangrove portal. The oncologist gets a clear answer: high risk or low risk. For low-risk patients, this could mean sparing them from unnecessary chemotherapy, months of grueling side effects, hair loss, nausea, immune suppression, all for a cancer that was never going to spread anyway. For high-risk patients, it means intensified monitoring and more aggressive treatment from day one, when it can actually make a difference. The tool could even optimize clinical trials, selecting the right patients, reducing the number of volunteers needed, and getting life-saving treatments to the people who need them most. Now, before you get too excited, there are important caveats. This is published research, not an FDA-approved device yet. The path from journal article to clinical deployment takes years: regulatory approval, integration with hospital systems, clinician training, insurance coverage. Most breakthrough AI tools in healthcare never make it that far. But what makes MangroveGS different is its foundation in fundamental biology, not just pattern matching, but understanding why cancer spreads. That kind of foundational insight tends to endure. For 90% of people who die from cancer, the killer isn't the original tumor, it's the spread. If we can finally read the biological program that drives that spread, if an AI can look at a tumor's genetic signature and tell us whether it plans to travel, then we're not just treating cancer anymore. We're finally beginning to understand it. And that might be the most important breakthrough of all.